Connaissance,
recherche,
sciences,
sciences mathématiques,
théorie,
physique,
philosophie,
Conférence,
archéologie,
nature.
So thank you very much to the organizers to invite me to give a kind of host, human host genetic view of what can we learn from nature. And basically what we are doing and what I will try to show you is how the human evolutionary genetics approach can contribute to a better understanding of human immunology. And I will show you how our research in the very end has not a lot of merit, because we are basically just unmasking what nature has already done for us, that is, select those genes or those gene families that plays a major role in host defense. So this is the tool of our research, is genetic diversity between individuals, between populations. And if you take two individuals randomly in the population, whatever their ethnic or geographic origin is, you will find very few genetic differences, what of course abolished, scientifically at least, the concept of race. But still, these very few differences in a 3 billion DNA basis of a human genome remains an average of 3 million differences in terms of changes in the genome sequence. An important thing to keep in mind is that most of these variants that we call neutral mutations have no phenotypic effects, both in terms of quality or quantity of the protein, and the distribution of these variants is just dictated by what we call in population genetics genetic drift, or what is basically random, and a human demography. So the distribution of this neutral variation, these neutral mutations, the study of that is very useful to infer human origins and migrations. But some of these genetic variants will have an impact on the phenotype, and some of these genetic variants are responsible for our phenotypic variation. Phenotypic variation that can be benign, that is simply our different physical appearance, but some of this genetic diversity can also be responsible for other phenotypic variation, such as the different ways we are going to metabolize food, milk, alcohol. Some of this genetic diversity is responsible for the different way we are, for how unequal we are in terms of response to therapeutic treatment, and we get into the field of pharmacogenetics. And some of this genetic diversity, for sure they are, and that's a field of research that is quite not mature. Some of this genetic diversity may be responsible for our different ways we react against vaccines. And some of this host genetic diversity is also responsible for our different reactions against infection, and also once infected, some of this host genetic diversity may also dictate the different ways we are going to progress to infectious disease. So this is more or less what we know in terms of our history, the history of our species. We left Africa around 60 to 1,000 years ago to colonize the rest of the world, but we had to adapt. We had to adapt to different environments, climatic environments, nutritional resources, but also we had to adapt to different pathogens, spatially and temporally. So the basis of our work is basically two keywords: human adaptation and natural selection, in the context of pathogens and infectious diseases. So genetic variants of the host that confers an advantage to be better adapted to a given pathogen will be naturally selected. Infectious diseases, as you know, have been a tremendous selective pressure over history. This is just a cartoon of survival mortality rates, where you can see, for example, that in the UK, in the end of the 19th century, here in yellow, around 38% of the population arrived to the age of 40, while today, also in the UK, more than 99% of the population arrived to the age of 40. While in many other countries, mostly developing countries, also here, for example, is in Mozambique in 2000, around 55% of the population reached the age of 40, what again highlights how infectious diseases have been an enormous selective pressure, but how infectious diseases are still an enormous selective pressure. The relation between natural selection and infectious disease is not something new. Already Allison in the 50s realized of the overlapping distribution of red cell disorders and malaria, what led eventually to the discovery that if you are an heterozygote for the HBS allele, you are better protected against malaria, whereas if you are a homozygote for the HBS allele, you will develop sickle cell anemia. This is another interesting example of how a loss of function mutation, that is the null allele in the dark locus, have reached very high frequencies in Africa, reaching almost fixation in West Africa, because it imparts almost complete resistance to vivax malaria. So all this to tell you that infectious diseases have been a major driving force in the evolution of our genomes. Just to remind you the different types of natural selection, this is the past, this is the present, these are chromosomes, and in blue you have neutral mutations. In red you have a mutation that confers you selective advantage, let's say imparts high protection against malaria, so over generation this mutation will increase in frequency in the population by positive selection. We have purifying or negative selection is basically the opposite. You have here black mutations that confers a selective disadvantage, higher susceptibility to a given disease. So over generations there will be the tendency to get rid of these mutations. And finally we have balancing selection, is a form of positive selection. It's not anymore the red allele that confers a selective advantage, but is the heterozygote state of this mutation, the red and the yellow, that will increase in frequency in the population. So today, for some genes, you will have an excess of heterozygosity. A textbook example of that, again, is the HBS allele in Africa. So what are we doing, which are our interests? Basically, as I was telling you, the keywords is natural selection and immunity-related processes. By immunity-related processes, I mean all what's related to host defense, immunity sensu stricto, or immunity at large. So we'll tackle these problems using genome-wide approaches to identify immunity-related genes being under selection in the genome. Why do we care about genes that are under selection? Because genes that are under selection are have played a major role in host survival, and there's lots of chances that still play a major role in our immunity to infection. We're also tackling these questions using candidate gene approaches or gene families approaches, and we are basically focusing on innate immunity receptors. I will give you an example of the toll-like receptor family, but we're also concentrating our efforts on other families of pathogen recognition receptors and their signaling pathways. The objectives being trying to distinguish, using this evolutionary approach, genes that play a major non-redundant role in host defense from those that plays a rather redundant role in host defense. And of course trying to put these evolutionary and population genetic aspects of what we are doing in a more general epidemiological and clinical genetic aspect. So one of the first questions that I would like to share with you is that we know that we have lots of phenotypic diversity among us. We've seen phenotypic diversity that can be benign, but also phenotypic diversity that can be related to disease. So one of the questions that we addressed 2 years ago was to which extent the phenotypic variation that we observe in modern human populations has been accumulated just randomly, that means by drift, or to which extent the phenotypic differentiation that we observe in modern humans populations has been helped, has been driven, at least partially, by natural selection, by adaptation of different populations to their environments. And if this is true, which are the main genes that have been targeted by this local adaptation, therefore positive selection? So for that, just let's try to come back before the differentiation of modern humans. So we'll come from an ancestral population in Africa. Or imagine that there was a mutation here in red that was neutral. So it was the split of the ancestral African population in what eventually become modern Africans and in what we eventually become modern Europeans. So if this mutation is neutral, the frequency of this mutation, this is the past, this is today, the frequency of this mutation will fluctuate by genetic drift. That means randomly. So for most of the genome today, the distances between populations for a given mutation are extremely low. The distances between modern human populations, genetically speaking, are extremely low. But imagine that we are again in the ancestral population, but in this case this mutation is not neutral, but this mutation protects against a given disease, let's say malaria. So there's the split into Africans and Europeans, but in this case there's a difference, and it's the selective pressure. So let's imagine that there's selective pressure in Africa, but not in Europe. So we come till today, of course this is a caricature, but this mutation will increase in frequency in Africa tremendously because it's useful. It protects against malaria. Whereas in Europe, it will remain at very low frequencies or even disappear. So for this particular mutation, the genetic distance between modern Africans and modern Europeans will be huge, maximal. That's just a statistical measure that goes from 0, that means no difference, to 1, that means maximal difference. So what we've done is we've measured the levels of population differentiation, so genetic distances, of around 3 million SNPs of the HapMap International Consortium that's made of 4 populations, 1 African population, 1 European population, and 2 Asian populations. And what we observed is that there is an excess of highly, extremely differentiated SNPs, so mutations between populations, that are particularly SNPs that do change the amino acid sequence of a protein, or SNPs, so SNPs means single nucleotide polymorphisms, that are located in the 5' untranslated regions. So these are SNP classes that present an excess of highly differentiated SNPs. An FST, this is a statistical measure of more than 0.65, is huge in humans. So the next question is where these mutations are located. Which are, in other words, which are the genes that are extremely differentiated between modern human populations? So this allows us to identify around 600 genes, where here you have a summary, that are extremely differentiated between human populations. Some of them are involved in morphological traits, what's nothing new at all, because we do know that there is phenotypic variation between populations, but it's a good control that the method is working. Other genes are involved in immune response to pathogens. Other genes are involved in a very interesting syndrome, that is the metabolic syndrome that includes obesity, diabetes, and hypertension, a syndrome that we do know that varies between human populations, regardless and corrected by socio-economical factors that do influence, obviously, the metabolic syndrome. And most interestingly also, a huge number of genes or yet unknown functions that are extremely differentiated between human populations and that may explain phenotypic differences between individuals and populations. More recently, we've done here a much more immunity-related meta-analysis of around 20 genome-wide scans for selection, where we have reassembled all the immunity-related genes that are consistently found to be under positive selection among human populations. We found genes involved in the interleukin-1 factors, interferon-related genes, NOD1, and other. In other words, all these genes is like a map of positively selective genes involved in immunity that may explain immunological differences between populations. We have also observed, when we can give a date to the selective events, that these immunity-related genes have been particularly targeted by positive selection in the last 30,000 years, suggesting that the last Neolithic and early and late Paleolithic period in humans has been very important for challenging our immune system. Also, let's go from genome-wide to a very anecdotal example. The evolutionary genetics approach can be very useful to address immunological debates. Here is an example of something that we did several years ago on the role of the mannose-binding lectin. The mannose-binding lectin is very known because there are 6 mutations, 3 in exon 1 and 3 in the regulatory region, that leads to a very famous immunological deficiency, that is MBL deficiency. But the paradox is that these mutations that are associated with MBL deficiency or low levels of MBL are found at very high population frequencies. So there's 2 ways to explain that. One way is that being MBL deficient has otherwise conferred a selective advantage. That's why these mutations are at high frequencies in the population. So in that case, the evolutionary expectation is that alleles and haplotypes associated with low or null MBL deficiency should exhibit signatures of positive selection. The other scenario is that basically we don't care if we are MBL deficient. At least we don't care evolutionary speaking. So that's why, in this case, mutations that are associated with MBL deficiency can freely increase in the population because do not change population fitness. So in that case, the evolutionary expectation is that we should not find any signature of natural selection. That is, that MBL should evolve under perfect neutrality. So what we did is that we sequenced MBL in different populations worldwide, basically using the Human Genome Diversity Panel of Ceph, and here you can see that deficient haplotypes in blue and low protein haplotypes in green can reach very high population frequencies everywhere. So we apply a number of statistical classical population genetic tests to detect the action of positive selection, and to make a long story short, this gene behaves as a perfectly neutrally evolving genes. More specifically, we also use linkage disequilibrium approaches whereby we can directly address the question of, do the 3 mutation or the 6 mutations associated with low or complete MBL deficiency have been targeted by positive selection? So the answer is no. So at least an evolutionary answer to the immunological debate is that from a population genetic standpoint, MBL2 variation has been shaped by strict neutrality, that haplotypes encoding for deficient or low MBL producers have not been targeted by natural selection. So our evolutionary data rather support a redundant role of MBL in host defense. That means that evolutionary and historically speaking, we don't care so much if we are MBL deficient. However, this does not exclude that MBL may play a gene modifier role in situations of coexistent morbidity, if you are going through chemotherapy or if you are already immunocompromised. In that case, being or not MBL deficient may play a role. One example that I want also to share with you is an example at the level of a gene family, the toll-like receptors that have been previously mentioned today by Stanley. The human toll-like receptors, well, in humans we have 10. 6 of them are mainly expressed on the cell surface and are mainly involved in the recognition of products from bacteria, parasites, and fungi, whereas we have 4 that are mainly restricted to the endosomal compartment, that are specialized in the sensing of nucleic acids, mainly from virus, but also from bacteria. So here the question we wanted to address is that how redundant or essential are toll-like receptors in host defense, and among the 10 of them, is there differences in terms of essentiality or redundancy in host defense? So to this end, we use the classical population genetic approach. We sequence the 10 toll-like receptors in different populations worldwide. And what we could observe first is that there's a distinction between a group of toll-like receptors that are under a strong purifying selection, whereas the other toll-like receptors, there's a relaxation of the selective constraint. What does it mean? Toll-like receptors 3, 7, 8, and 9 are under a strong purifying selection, what means that these 4 toll-like receptors cannot accumulate mutations at the population level that do change the amino acid sequence. Whereas there's a certain freedom for the other toll-like receptors to accumulate mutations that will change the protein. This dichotomy is interesting to see that perfectly correspond to the 4 intracellular nucleic acid sensors with respect to the cell surface express TLRs, suggesting that either viruses or products that are sensed through their nucleic acids have exerted a stronger selective pressure on the host, at least from regarding the toll-like receptor systems, or other forces that may have contributed to the extreme conservation of toll-like receptors. Intracellular toll-like receptors is autoimmune avoidance, but we can discuss this in the questions. Another interesting observation, here you have the proportion of individuals in real life, that means the general population, that present at least a mutation that changed the amino acid sequence and considered to be probably damaging in yellow, or even a stop mutation that truncates the protein. So you can see that for intracellular nucleic acid sensors, they cannot allow to accumulate this kind of mutations, whereas for the remaining toll-like receptors, the cell surface express, there's examples like, for example, toll-like receptor 5, where the dominant negative mutation is present in 12% in that case of the European population. What this suggests? This suggests a certain redundancy for toll-like receptor 5, a certain duality maybe in the recognition of the innate immunity systems for flagellin in this particular case. But the higher flexibility of the cell surface expressed TLRs also allowed them to accumulate in some cases mutations that can confer a selective advantage. This is the case for a particular haplotype that we have identified that encompasses toll-like receptor 1, 6, and 10 that has been under a strong positive selection in Europe. Unfortunately, these three genes are one near to the other, so the only way to see which is the gene that is responsible of the patterns of positive selection and which is the functional consequence of this selective advantage were some functional studies that we did, and what we showed is that of the 3 genes is toll-like receptor 1 the responsible of the selective advantage and of a particular mutation that decreases but at least half the percent of NF-kappa B activation, used of course as an in vitro proxy of the immune response TLR1 mediated. So to finish with this almost last example, what we show is that toll-like receptor 1 harbors a mutation that involves a diminished immune response that has been a selective advantage in Europeans. So this raises question about how a diminished immune response can confer a selective advantage. What's interesting to note is that the same mutation has been associated with protection against tuberculosis and leprosy, that are infectious diseases, as you know, but are also inflammatory diseases. What raises questions about the good compromise that innate immune response has to reach between a proper recognition of the microbial product, but also avoiding too much inflammation that can be harmful for the host. And the last 2 minutes example is a more recent work that we have done with our colleagues of epidemiological genetics in Pasteur, where what we wanted to do together was really to intermingle and to integrate the interest of an evolutionary genetics approach and an epidemiological genetics approach. In terms of glucose 6-phosphate deficiency, it's known that a deficiency has been associated with protection against malaria in Africa, and the allele that confers the deficiency that is associated with resistance to malaria in Africa has been dated to around 3,000 years in Africa. But the point that we were interested in Southeast Asia, where there is another G6PD deficiency variant that is called the Mahidol variant that exists at very high frequencies. 10 to 25% of Southeast Asians, particularly in Thailand, present this deficiency variant. So the first question we wanted to address was how being G6PD deficient in Southeast Asia has conferred a selective advantage. First, has it conferred a selective advantage? So the answer is yes. I avoid you all the technical details, but what we could show is that being G6PD deficient in Southeast Asia has involved a huge selective advantage. We could even date the date at which the selective advantage started, and we dated at 1,000 and 500,000 years, with intervals of confidence that are quite good, actually. It's between 1,000 and 3,000 years ago. We also measure how much this has conferred an advantage, and I'm not going to get into any detail, but the selective coefficient of this mutation, that means how good has been for the host, is one of the top 5 of the human genome. The next question was, of course, which is the selective advantage associated with the strong patterns of positive selection of this mutation. So for that, our colleagues did a very long and tedious epidemiological survey, and what they show here is the parasitemia of Plasmodium falciparum density, what's known to be influenced in Africa for G6PD deficiency, another mutation. And here in blue you have the wild type, and in kind of orange, individuals that are G6PD deficient. So being G6PD deficient does not change parasitemia in terms of Plasmodium falciparum. But the surprise was that being G6PD deficiency decreases significantly the parasitemia of P. vivax in Southeast Asia, showing that if you are G6PD deficient in Southeast Asia has been under a strong positive selection for the last 1,500 years, since it reduces P. vivax parasite density in humans. This raises questions also of how the introduction of rice culture in Southeast Asia, coming from China, that was at that time, increases also the exposure of people to vector-borne infectious diseases. So, to finish, I hope I was clear enough in showing the interest of bridging the gap between evolution and medicine. You know, with this kind of tools like diversity, we can address questions such as, has this mutation a phenotypic effect? That is this mutation associated with a better survival against malaria? Is this mutation associated with a better reaction against the vaccine? And here we use medical and epidemiological genetics approaches, but also we can address question of when this mutation appear, or why this deleterious mutation involving an immunological deficiency is present at such a high frequency in the population. And for that we use population and evolutionary genetics. The idea is one approach informing the other and the other way around. So just to thank the people that have been involved in all these studies, in Pasteur, in my team, and our collaborators and funding agencies. Thanks so much.